Go on in the program. We have one talk more before the break, and the talk will be given by Peter Pöchlauer. Peter is Innovation Manager at Thermo Fisher Scientific. He studied organic chemistry and pharmaceutical chemistry at the University of Innsbruck, Austria. And if you know it, it's in the middle of the Alps, and he just told us they have snow over there, it's winter. Now, 2 years later, he made a postdoc under Professor Rolf Huisgen at the University of Munich. And that makes click with me, a kind of click chemistry, because that is a 2+3 cycloaddition. He's so famous for it. He's like the hack of this kind of reaction, so very famous name. He has more than 30 years of experience in pharmaceutical research and development in the industrial sector with Thermo Fisher Scientific. Between 1994 and 2004, he developed several large-scale biocatalytic processes for fine chemicals and led European research projects on biocatalysis and sustainable oxidation chemistry. From 2003 to 2006, he headed process technology and introduced process intensification and microreactor technology in the company. In 2007 to 2009, he helped to introduce one of first continuous flow processes in large-scale pharmaceutical manufacturing. I remember that gave a huge push to the technology, especially in Europe, because Japan was much in advance at that point of time, and we needed good examples in Europe. So presently, Peter is working to drive innovation in small molecules manufacturing to develop cleaner, faster, less wasteful, and more efficient production processes. Peter, we look forward to your talk, which is entitled Towards Digital Twins of Intensified Processes.
So, Oliver, thank you very much for this extensive and very kind introduction. I'm trying to share my screen now. So, I think this is, I hope everybody can see my screen now. And yes, as indicated by Oliver, it's about digital twins of intensified processes. And I think it's best if I start by explaining what the talk will be about. First, of course, who after all is Thermo Fisher Scientific, a company that everybody knows is producing lab chemicals and lab equipment such as chromatographic systems and thermocyclers. This company has learned to manufacture chemicals in custom manufacturing campaigns. Now, for which clients do we do that, and what are the priorities of our clients? What are the secrets of active ingredient process development to serve these clients, and what's new, what's different? What's complex in this business at that moment? Is it the chemistry? Is it process technology? Is it the market? I'll talk about that. And then what role does process intensification play? How does it link to advanced continuous analytics? And how does it link to advanced mathematics, as I would call it? So let us start with Thermo Fisher Scientific. So we call ourselves the world leader in serving science. What does this mean? We don't think we're the best scientists, but we think it's very necessary to put our scientists in a position where they can live up to their scientific aspirations. So it all started with Thermo Scientific, applied biosystems, in vital change, a lot of molecular biology, and lately a unit called Patheon was added. Patheon was actually known for its formulation capabilities, and to that was added a number of manufacturing facilities and sites that were capable of manufacturing not only the finished tablets, but also the active ingredients. So Patheon had a strong drug product, and to that was added TPX. This is where I came in, and American small-scale sites, large manufacturing sites from Roche, and lately also from GlaxoSmithKline. And in doing that, Thermo Fisher, presently a company of more than 100,000 people, has acquired capabilities of developing and manufacturing pharmaceuticals. I'll talk about exactly this tiny edge of the huge Thermo Fisher. First, a question from outside in: who, after all, developed new medicines? And you would say pharma companies. From the press, we learn that the new molecular entities that were originally developed by small, mid-sized, and big pharmas has changed. 12 years ago, small pharma was responsible for only a third of new molecular entities, and 3 years ago, we had also already 3/3 of all new molecules being developed by very small units. And another voice from the press: these journalists found that large pharmaceutical companies did not actually invent most of the drugs they sell. Indeed, they have already reduced their investment, and what are they proud of? They focus, of course, on taking these things to the market, taking these things to the bedside, and distribute them in a flawless way. And again, in 2020, we of course do those market research activity. 10 of 35 small molecules were launched by large pharma houses, a third only. And how do large pharma houses get their new molecules? They acquire projects of small companies in late stages of development. For example, we have active pharmaceutical ingredients that already during development change hands several times on their way from the research laboratory, in this case Scripps, via small companies, bigger companies, very big companies such as Bristol Myers Squibb. And so it appears obvious that we have to know the needs of our clients very clearly. Let us look at the speed of development. We see that a continuously growing fraction of new drug candidates are developed along expedited pathways. So what does this mean? These are opportunities offered by the regulatory authorities for special, much needed drugs. So you have about 60% of the novel drugs were approved to orphan diseases. That means diseases that only a very small number of patients suffer from. So large pharmaceutical manufacturers would not be in a position to regard them. So there's a specific push by the authorities to have even such medications developed. And you see that this has risen from 44 to close to 60% orphan diseases. Or you have novel therapeutic concepts called first-in-class medications. 40% are first-in-class, so not the umpteenth ACE inhibitor, but completely new therapeutic concepts are pushed by the authorities. Or likewise, for example, breakthrough therapies, 40% breakthroughs. That means therapies for patients that did not have access to appropriate therapy up to now. And of course, you can also ask for priority review. If you think that the world needs your medication very quickly, you can ask for priority review. That comes at a cost, but it's very efficient and helps the FDA to keep their high quality standards. So that has also risen. So all in all, you have more than 2/3 of all newly developed medications that use one or more expedited pathways. So what does this mean? What does this mean to everybody who develops and later manufactures pharmaceuticals? First of all, one could say, okay, orphans, we have more drugs for less patients. Yes, this is the case because the majority of patients, those suffering, I would say, high blood pressure, they have perfect medication. You have more drugs for less patients, but this opens a pathway to personalized medicines. Definitely, the industry learns to react to needs of small numbers of patients. And it also means, of course, more projects, faster projects, and only a few large projects for which we have fierce competition. So, of course, this adds a lot of complexity to that business. But where does this complexity come from? I would say, first of all, we mentioned that despite the spectacular success of large molecules, we still have 2/3 to 3/4 of all new active ingredients are small molecules. So a chemist's business, and distinctly different from, for example, microbial fermentation. And most small molecule pharmaceutical lead compounds are first synthesized, as we all know, in libraries. The applied chemical synthesis methods are optimized to tolerate the substrate variability. So not manufacture one single molecule at optimum efficiency, but to be tolerant to high substrate variability. And consequently, in many cases, these methods are not ideally suited for scale-up. It was not their initial reason to develop them. Secondly, still the structures are simple. We see hardly any large numbers of chiral centers, and we have a comparatively small number of standard transformations. The Suzukis, the Hecks, the Buckworths of these words, if you look at the structures, they might be comparatively big structures, but actually they are flat. They are a number of cycles or heterocycles linked to each other using standard linkers such as amides or aromatic coupling techniques, and so on and so forth. So the complexity in development projects relates, next to the speed, to the physics. You have to take care of physical properties of substances. You have to be in control of a polymorph, and parallel to development of the synthesis, you have to develop the formulation. So all of a sudden, you have a chemist in need to understand the physics of what he does. So if you look at our typical business processes, how do we serve our clients? So this is admittedly a clumsy synthesis of a medication, of a prostaglandin that indeed we manufacture. And a company developing a new medicine asks us to produce an intermediate or the whole synthetic route of an active pharmaceutical ingredient. So we go down all the way. So, and in discussions, we start asking them, do we have an idea of the volume forecast? How complex is the chemistry? What is the competition? What is the risk that this thing will fail? What are boundary conditions? What are timelines? Of course, does it need to be quick or very quick? And then we take a decision, yes, we embark on this project. And then we do this translation of a library synthesis into a scalable synthesis. And this is the first point where technologies kick in. To do this efficiently, you need new technologies. And then we enter process development, scale up, we do safety considerations. What would a plant look like? What would the investment be? And this is the second stage where new technologies kick in. So, and then we deliver the material, and then we wait for the result of clinical trials. And therefrom, we learn, is there a further demand? What would the future volume be? What would the future price competition and so on be? And once the product lives, we continue and reiterate this process. So we develop the synthesis further, we develop the technology further to serve whatever volume and quality needs our clients might have. So this is where we earn money. What are the installations we use to earn that money? Actually, manufacturing of pharmaceuticals is very conservative. Traditionally, we have continuous manufacturing of physical parameters. You all know this: temperature, pressure, weight. We have a continuous monitoring of very few chemical parameters. This is where we come from, yeah, pH. We operate in batch-wise production, and we monitor and control by taking samples. You have a horseman taking the samples from manufacturing building to the analytics building, putting this sample into a queue, waiting to be analyzed. we have samples for information to see whether everything is online, for monitoring in process controls, and we analyze them. Analytical results are, of course, valid for the analyzed sample volumes and for the time point when the samples were taken only. The analysis time, and this is a horror, may significantly contribute to the cycle time of a process. And this is, of course, something to be avoided at all cost instances. So even if you create a number of data, what you see is you have the physical parameters, the few chemical parameters, they come directly from the plant. They come from the analytics lab. And these data are all stored, at least in most large companies, in different formats and at different storage locations. So therefore, and seeing and suffering from all this, we have started programs to apply in an opportunistic way, always to pay off, concepts of continuous processing in small structured reactors. We have started to increase the number of complexity of chemical parameters to be continuously monitored. So what's in your reaction mixture, at which concentration, what is starting material, what is product, what is byproduct, and we use the generated data to improve our process models and to improve our process understanding. This is what the authorities ask us for. Are you in control of your process? Do you understand your process? How do you learn that your process is going straight? Now, we have seen many of these pages before. I just reiterated it. We have purchased or custom built, homemade. I even built a couple of reactors with my own hands and used different reactor types. So structured plate reactors in Hastelloid and structured glass plate reactors, structured plates in silicon carbide, custom built stainless steel tube reactors, or 3D printed metal reactors. And these are only the reactors I am allowed to talk about. Of course, being in close collaboration with our clients, many clients would not like us to talk about the way their materials are produced. So is now continuous manufacturing well established in large pharma houses only, or also in the CDMO industry? Let's look at the press. Again, here we have it from Sequence.com. More and more contract manufacturing organizations, and Thermo Fisher is one, are embracing continuous or flexible manufacturing alternatives. So that means this is no longer experts-only territory. It approaches mainstream. And the message to our customers is, of course, the equipment is operational to produce your material. Then many new therapies are receiving fast track or breakthrough, and we are challenged with significantly speeding up the chemistry. We have talked about that before. And of course, we can use new manufacturing technologies to meet these accelerated timelines. And we have shown that before. And then the FDA came out with a guidance to support flexible approaches to manufacturing. This is very important. Will my auditors like what I'm showing them? Yes, they will. Continuous manufacturing is encouraged by regulatory authorities, and the authorities even offer regulatory assistance. And last not least, the need for flexibility in manufacturing has never been greater, because new drugs can be more precise and powerful and have to be produced in smaller volumes. So you have orphans and you have highly active materials. So yes, continuous manufacturing intensified processes can handle unpredictable volume demand. Our partners usually do not know their volume demand prior to marketing their material. So continuous manufacturing can avoid shortages at the one hand and overstocking at the other hand. A quick look at the different chemistries we have taken to various degrees of maturity. It all started with a non-pharma production on very large scales. This is still up and running. Then we did a nitrate ester formation, low temperature methylation, acidation, ethyl diisoacetate, hydrogenations, high temperature hydrochlorination, chlorination, diazomethane, and these are only the ones, as I said, I'm allowed to talk about. And all these developments come with their analytical requirements. So the stepwise introduction of continuous analytics is a further goal of our process intensification program. So you usually follow a rationale where you start with a proof of principle in the lab. You create data on reliability and operability on larger scale. You validate the respective method. You use it for process control, and you even may be able to use it for quality control and release. So the proof of principle in the lab has of course been done for the largest number and variety of analytical methods. We have used NMR, FT, NIR, Raman, mid-IR, laser diffraction, FBRM, microscopy, and this list is, believe me, it's still growing. Then operability in the plant has been proven or shown through comparative studies of FT, NIR, NMR, and Raman, and we have validated analytical methods in the plant for FT, NIR, and FBRM, focused beam reflectance measurements for slurries. And FT, NIR has also been validated to be used for process control in several instances. So this is the path we're on. And as I've talked or mentioned before, let's compare what we are presently doing to what we should do. What we should do, and here I quote the Horizon Europe work program 21-22, and it comments in a first destination, as they call it, on an appeal for climate neutral, circular and digitized production. And what we read here is they would like to see data-driven distributed industrial environments. So data-driven, distributed, so no small number of large sites, but distributed sites, distributed manufacturing facilities. And in the second destination, even more clear, increased autonomy in key strategic value chains. So this is EU verbiage. And all that, of course, all that links to the sustainability and security of supply of essential chemicals. Now, we have reacted to that by establishing further collaboration programs. So all this can only be done with collaborations of industry, with research institutes and with universities. And I quickly give you an account of what we have lately been doing in a new, as we call it, competence center. And this competence center is called Chemical Systems Engineering, abbreviated CHASE. And it's all about comprehensive and holistic digitization in the process chemical industry. We start from varying feedstocks to produce defined product quality. And this group is doing research at the interface of chemistry, process engineering, and analytics. And I should add mathematics for industrial production technologies. So it has three pillars: process digitization, process intensification. This is all about what I'm talking now, and circular process streams, focus based on strategic programs to do basic research. And we already have a number of sizable companies that are partners to this. So from oil field to paper to polymers, to pharma supplies, to equipment supplies, and pharmaceutical manufacturers as Patheon. And from this collaboration, I give you an example of a process model, how we link that intensified process to digital models, to mathematics, to learn about it. We started by selecting a finished production campaign of a pharmaceutical product. Then the way was to define all potentially relevant data, make them available for the modelers, and correlate them by their timestamp. assign them to a specific production batch, condense the data into features. And what does this mean? A feature. So here you might have the readout of a temperature sensor. It gives you 20 to 50 to 1,000 points in a certain time span, and you condense these data by the period and the difference, and you replace it by a feature. Temperature rises by X grad Celsius within Y minutes. So you have two variables instead of 50 or 20 data points. Then further on, we established a data set characterizing one batch, cutting the number of variables down, and then exclude outliers, of course, and establish models for selected process outputs, so responses of these models, and then apply the models to watch what was going on in an actually up and running production batch. So apply the models as decision support system to operate and optimize the process. So let's give a graphic representation of what this means. So the preconditions, of course, data exchange, spaces, and so. And what you see here is a number of pieces of equipment: reactor 1, reactor 2, equipment A1, it might be a filter 1, a dry one, and their engagement over time. And as the starting material passed through the plant, passing from one piece of equipment to the other one, you turn them into product. And as soon as the first reactor gets idle, of course, you can refill it, and you can start another batch and another one and another one. And this is called one batch. Now, is it easy to follow this batch by data? Let's have a look at it. If you look at one process control sensor over a certain time span, it might sit in that apparatus A1, you get this signal. Of course, it's filled, it does something, it's emptied, and then this sensor senses some spike, and so on and so forth. On the other hand, at any given point in time, the process control system monitors, for example, up to 3 batches. You see that? 1 time step, 3 batches. And then on top of this come the shift workers plan. So at one point in time, this apparatus is operated by the blue shift. In the next batch, the same piece of equipment is operated by the red shift. And there is a handover to the green shift. So that has to be considered. So, and then you have in-process controls. So they give you a picture of a moment here or here. And what you then have is a bunch of data with very different sources: the process control system, the batch record, SAP, external factors that need probably be considered or not, the model will show us, cooling water, shift data, and many of these data are still in Excel tables. So data sources must be integrated, correlated. From the huge volume of data, you can define a standard batch. This is the golden batch to which you compare what you are presently doing. And the model, of course, must be tolerant to events. For example, you recalibrate the temperature sensor. And the modeling environment must be easily transferable to a different process. So whenever I use this reactor for a different product, I wouldn't like to start at 0. So this process model consists of various elements. First, the process. You're in a complex equipment. You have many unit operations, many batches. And you have a large body of very diverse data. And the goal is to have a process model capable of predicting the results of in-process control. So you're sitting on a large volume of data. You have a plant up and running. And your goal is to predict what is going to happen based on this data. And the task is to establish that make the data sources available, integrate them, do a statistical analysis, build a model, and design from it a workable tool for the operators. So here you have your plant, and this decision support system should give something like the following messages to the shift lead. It should tell him, based on the analytical data of starting material 2 that might have come in here, and based on the temperature in your vessel 1, and based on the results of the IPC1, so something like this here, your IPC2 result will be close to the upper limit. So taking all these data into the model for the up and running batch, you will get this message. And the message will go on, shortening the stirring time in vessel 4 by 15 minutes will move the result of IPC2 towards the target value. So this will keep you on track. And the way to reach this is, of course, as I said, make the process control system data available, extract the features, correlate with other data, integrate, and do a statistical analysis, because you're sitting on data of differently operated batches. So how is this statistical analysis done? Of course, first of all, you analyze for idle times. Where do we have delays? Why do we have delays? And we do first correlation analysis and trends over the whole campaign. A good way to do a statistical analysis is a principal component analysis. What are the principal factors that will affect a certain response of that model? That might not be a single factor, that might be a combination of factors. And keep in mind, one batch is characterized by something between 200 and 300 variables. So, and as we call it, the loadings of those principal components might be a number of parameters, others might be less important. And we start correlating those principal components to, for example, the idle times. And what you see is even a shift in color, with the blue color signifying the early batches with low shift numbers, and the later ones, the yellow ones, the later batches, as the shift workers have learned how to react to it. You see a clear tendency, yes? And from this, you can start asking yourself, what does this mean? Is there some systematics behind, and so on and so forth. And the second way we look at those models is, of course, we try to model the in-process controls to be able to predict them and try to explain the variance in in-process controls. And some of the in-process controls are already pretty well explained, and others are poorly explained in their variance. So we know that there are still factors or effects hiding behind what we have presently monitored. So it's very important that you see, okay, here I can explain what I'm doing. I understand my process, and there are IPC elements that I do not understand. There is a certain degree of uncertainty what will be, even if I think I'm in control of all this. And this is exactly the sort of discussions the regulatory bodies want me to enter with them in asking me, please prove to me, why do you think you understand your process? Now, our conclusions from that. The development and the continuous improvement of sustainable manufacturing process is a huge activity. It needs the tight collaboration of chemists, experts in synthesis and analysis, of process engineers. It's not to mix which chemicals, but how to mix them. Then plant engineers, in which piece of equipment to mix them. process control experts, how to monitor that mixing in the lab and in the plant. And then, last not least, the statisticians, the mathematicians, and they have to translate the language of their findings into a language that the shift operator will understand. Only if we can do that, this will allow us to meet the requests by our governments, as I said, climate neutral, circular, digitized production, and increased autonomy in key strategic value chains. I think that's been it. Thank you very much for your patience, for bearing with me. If you have any questions, I'll be happy to answer them.
Thank you, Peter. That was an excellent talk. We have a question from Josh from Molecular Boron. He said, great talk, Peter. The post-reaction workup is the often forgotten aspect of flow chemistry. That is very true. How many of these processes on a pilot scale are already worked up or purified in flow, or do most of the auxiliary equipment need help with the workup?
Very good questions. Actually, that's a separate talk.
Yes.
Even when we started in intensifying the chemical reactions because there was a need, we then worked our way through the process to work up that chemistry, that reaction. And if you consider the plant scale pieces of equipment that you use to work up your reactions mixture, you see, you have a mixer settler, you have an extraction column. All these are pieces that are best operated in a continuous way. You may have a distillation column. Of course, a distillation column is best operated in a continuous way. And you can also intensify all these things. You can replace the mixer settler by centrifugal extractor. You can even go for intensified distillation. And we have even worked our way to solids, liquids, handling and separation. So we do have, for example, continuous filters, belt filters on plant scale. And we have lately also established and set up continuous dryers, for example, fluidized bed dryers. So yes, a perfect question. Yes, very important to have a continuous manufacturing device in an environment of further continuous pieces of equipment to work it up.
Let me give a last question because we're over time. I'd like to condense two questions because they're very much related. One is from Tash. Again, fascinating talk. So he asked about the robustness and accuracy of your process model. And in a similar way, Bert from Ayanamoto asks, is it necessary to condense data? Data is a new oil. AI can handle complex data better without the human processing, or is it still too early for AI? I think those are similar questions. Maybe you can answer them together.
So my mathematicians still were very keen on reducing the number of data, but think of a manufacturing campaign that may take, I would say, 6 months, and you get temperature readings once in a second from 50 temperature sensors. So this is a wealth of data that needs to be structured. I think we would have the mathematical and the computational capacity to do that, but it wouldn't make sense. So what we do a very early pre-processing of the data to condense them. We have also found so highly correlated data that we can take out one set of data, of course. Yeah. So if you have two temperature sensors taking care of each other, because safety requires two sensors or probably two independent measurements, you can take them out of the model as long as they work. Okay. And that's why my mathematicians told me, please take them out, or we suggest to you to take them out.
Thank you. I think that was very insightful. Thank you so much for your talk. We are 10 minutes over time. Let me make a proposal. We make a 15 minutes break instead of 20 minutes, meaning we start 5 minutes later than the plan Well, that still gives you all time to relax a bit. And I'd like to announce already here that at the end of the whole summit, Christian will give some information on the next IMRET conference, just 2 or 3 minutes. So if you're interested in the IMRET, please stay online then. It will not be finished, this summit with the last talk, but just 3 minutes more. Okay, so we see in 15 minutes from now on. See you then. Bye-bye.